Okay. Okay, let's get started. Good morning, everyone. Welcome to CCMMP's monthly seminar. Firstly, I'd like to begin by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining me from today. I pay my respects to their elders, past, present, and emerging, and to the Aboriginal elders of other communities. My name is Dongju Lee, PhD student and Monash node of CCMMP. I'm chairing this seminar. This seminar will be recording and you will find on CCMMP website. So please make sure to stay on mute while our guest speaker is talking. At the end of the talk, we'll have a Q&A session. So if you have any questions, feel free to type your questions or raise your hand. Today's guest speaker is Assistant Professor Christopher Barnes. He's a newly appointed Professor of Biology and ChEM-H Institute Scholar at Stanford University. His group combined interdisciplinary approaches like biophysical method and in vivo systems to understand the way of virus host infection system, such as HIV-1 and SARS-CoV-2. So during the pandemic period, he has over 15 pandemic-related publications. Now I hand over to Christopher.
Thank you so much. And it's a pleasure to be with y'all in the morning there and evening over here. So let me share the screen. So today I want to talk to you about some of the work I've been doing the last 2 years, mostly as a postdoc, but I will highlight some of the new work that I've done in my own lab, related to SARS-CoV-2. So I first want to highlight, like I said, that a lot of this work was done during my postdoctoral fellowship in Pamela Bjorkman's lab at Caltech, mostly in collaboration with Michelle Newson's lab at the Rockefeller University. So I worked with a great number of students and staff scientists, but I really want to highlight Beth Huey-Tubman, who was my technician, as well as Claudia Jetty, whose work you'll see today. And from Michelle's side, Davide Robbiani and Vinci Wang, who helped characterize some of the very first antibodies. So back in 2019, I like to show this. This report was commissioned by the WHO, talking about are we prepared for possible spillover events of newly emerging and re-emerging viruses. And they highlight in this report that was published in September 2019 that there's a ton of viruses that could lead to devastating epidemics or pandemics that cause a huge loss of life as well as upend economies. And then, of course, just a few months later, then especially within Western society, in March 2020, we saw this huge outbreak of SARS-CoV-2. So as we know, SARS-CoV-2 is a current pandemic from which we're still living. Despite effective vaccines, we're still seeing in the hundreds of thousands of new cases reported each day across the world, with many thousands of deaths across the world as well. Right, so understanding SARS-CoV-2 requires multidisciplinary approaches, not only in prevention, but also in therapies. And so SARS-CoV-2 is an RNA virus. It's actually the coronavirus family is the largest family of RNA viruses, with the RNA genome of around 30,000 base pairs. So there's an envelope virus, RNA virus, that has a spike glycoprotein on the cell surface, which attaches to the host cell by engaging the receptor called ACE2 on the host cell. On the host cell. This attachment then leads to either a fusion event at the plasma membrane between the host cell and the virion, or endocytosis, and then fusion between the endocytic vesicle membrane and the virion membrane, releasing RNA, which is then translated into new proteins, which are then packaged and released to go and infect new hosts. So neutralizing antibodies, if you think about them, are our body's way of defending ourselves against these viruses, and that they can attach to this spike glycoprotein and basically mediate them from being able to enter our host cells. And so if we take a zoomed in approach onto a single SARS-CoV-2 variant shown here through this tomographic reconstruction, you can see that the spike glycoprotein shown here in red in this pink or salmon color decorates the surface of the variant quite densely. There are two integral membrane proteins that are also on the surface of the variant, but aren't the main targets of our neutralizing antibody responses. Then within the variant itself, you have the packaged RNA. Right, and so the spike glycoprotein, as you can see, kind of sits here and it was really exposed, and therefore it's the main target of our neutralizing antibody responses, because in essence, this spike glycoprotein is what the virus needs in order to hook on to the surface of our cells. And so if we look closer at the spike glycoprotein, we can see that it kind of exists in two distinct conformations. One conformation shown here on the left, where you highlight the S1 domain, which is here in the apex, and the S2 domain, which has an integral membrane component, as well as a cytoplasmic tail. The S2 domain is responsible for the fusion machinery, whereas the S1 domain is responsible for attaching to the host cell ACE2 receptor. And so if we focus in on the region that actually binds to the ACE2 receptor, known as the receptor bind domain, or the RBD can exist in two kind of local conformational states. One being a completely closed state where the receptor binding motif is unexposed, or you have this up state shown here by this one RBD, which now the ACE2 receptor binding motif is exposed, allowing attachment. Now, due to its relevance in attaching to the host cell receptor, the RBD was the main target of most of our neutralizing antibody responses. And of course, from many studies, including our own, we have shown a correlate of protection with the more higher RBD specific titers in serum compared to other antibodies targeting different domains. So given that we understood that antibodies are our main defense against SARS-CoV-2, we wanted to begin to understand what could actually be utilized as therapies. Right, so at the very beginning of the pandemic, before we had any known therapies, the idea of using convalescent serum was really our only way we could treat patients that were showing up in the hospital. So within this convalescent serum, you have a polyclonal mixture of antibodies, some with specificities to the RBD, to the NTD, to the spike protein, to other parts of the variant, all displaying different levels of potent neutralizing activity. And then within this mixture, you have kind of these defined monoclonal antibodies, right? And so these are the antibodies, that have high specificities, so maybe the RBD, are very potently neutralizing, and are the main target when you're starting to think about making new antibody therapies. So the main point of the work idea as a postdoc, and now currently as an assistant professor, is kind of trying to understand what are the correlates of the antibody-mediated protection from SARS-CoV-2. So all this work was done in the background of our work in trying to understand the same types of antibody-mediated protection against HIV-1. So we were really situated to understand this phenomenon during this pandemic. So the first thing we want to look at, in quick snippet of stories that we've published over the last year and a half. The first story was actually looking at the polyclonal response, because again, we understood that people were utilizing convalescent sera to treat patients, but from our collaborators, Davide Robiani and Michelle's group, we knew that there were different levels of neutralizing titers from convalescent donors. And so these donors were people that were convalescent within 1 month of being negative for the virus after recovery, or I should say 1 month post-symptom onset after being infected with the virus. And so we could see that some of these patients, like number 47 and 21, had very high dilution titers, suggesting that they had very potent neutralizing activity within their sera, whereas individuals like 107 had very undetectable or very low neutralizing activity, despite the fact that they recovered. So we knew there was a range of responses for neutralizing activity among people that were infected. So using this technique called negative stain EM, polyclonal epitope mapping, or negative stain MPEM, I took people's convalescent sera from different donors, cleaved their plasma IgGs into Fab fragments, and then took those Fabs and bound them to the antigen, in this case, spike glycoprotein. And what we're able to see from these studies is that people that show really high correlates of protection or really high neutralizing activity tend to have antibodies targeting the apex, where we know that receptor binding domain kind of resides. whereas people kind of in the middle tier show, different epitopes being targeted, including those in the N-terminal domain, like this one shown here. And the individuals like 107, didn't have really any dominant antibody response circulating within their system. Now, it didn't mean they didn't have, antibodies that were able to neutralize the virus. It's just that the main neutralizing or circulating antibodies within their sera were, not dominant towards any specific epitope that could be detected with this technique. But from this study, we first, began to realize that we have two kind of main targets of the neutralizing antibody response. One to the receptor binding domain shown here at the apex, and one towards the N-terminal domain shown here in green. And so that led us to begin to focus in on now these receptor binding domain specific antibodies. And so we turned to a more high resolution technique, single particle Cryo-EM, with defined monoclonal antibodies that have been cloned from circulating memory B cells. And so in Cryo-EM, for those who are not familiar, what I'm showing you here is a representative micrograph of a spike glycoprotein. These kind of squiggles represent a spike glycoprotein bound to two Fab fragments. We can then, extract these particles from these micrographs, average them in 2 dimensions to begin to appreciate the different angles and orientations of the spike protein complex within this micrograph. So you can see here kind of these side views. We have the spike glycoprotein density, and then you have this diffuse density at the top corresponding to the Fab being bound. You have these top-down views. So as long as you get enough of these views of your protein, you can then take that and create these 3-dimensional reconstruction volumes, which you can then model into these volumes, coordinates in order to understand exactly the molecular, the molecular insights into your structure, into your complexes. And so what's wonderful about Cryo-EM, of course, is that it's, even though it's a state, it's a frozen sampling state, you can actually image many different populations within your sample because these samples are in solution before being frozen. So take this example here, where we can kind of tease out 2 representative classes, one being a spike glycoprotein in which 2 RBDs are in the up conformation, one is in the down conformation, and then spike glycoprotein shown here where all 3 RBDs are in the up conformation. And what we could tell from this was that in the case of this antibody C105, the up conformation was the only conformation that the antibody could recognize, suggesting that this antibody, epitope was only exposed when the RBD, flipped up, similar to what we see with ACE2. And so from this study, that was published in Nature in 2021, we were able to identify, or say in 2020, we were able to identify 4 distinct classes of RBD-specific antibodies that are termed class 1, 2, 3, and 4. With classes 1 and 2 overlapping with the ACE2 receptor binding motif, which we sit here at the apex of the RBD, shown here in gray. Class 4, only being able to recognize this epitope. This is a highly conserved epitope that we'll come back to, but it's only exposed when the RBD is flipped up into an up conformation. And then class 3, which sits outside the ACE2 receptor binding motif, are actually very potent, also targets a highly conserved epitope that we'll come back to. From these different classes, we were able to also identify potential escape mutations for each class shown here, which allowed us to first identify with our collaborators that E44 mutation as a site of vulnerability for the class 2 antibodies, which are the dominant circulating antibody class found within convalescent donors. So now that we had this classification, kind of understood the makeup of antibodies and their characteristics, targeting RBD from convalescent donors, we actually questioned, well, is this the same among people that are vaccinated? And so as the vaccines roll out, especially mRNA vaccines here in the US, we took vaccinated donors and compared their neutralizing activity, seroneutralizing activity, and showed it was very similar to those that were convalescent after 1 month. But then we asked the question, right? Is a spike glycoprotein that's being expressed on the surface of our cells and now being targeted by our immune system, recognized by our immune system, is it functionally and structurally similar to that, of the spike glycoprotein on the surface of a virion? And so we cloned antibodies in a similar fashion from vaccinated donors who were naive to actually SARS-CoV-2 infection. And then we took a random sampling of these antibodies that were RBD specific and did structural and characterized these structurally again. And what you see is sampling against the RBD epitopes, very similar to what you see in convalescent donors. So those that are class 2, class 1, class class 3 and class 4. And so at the end of the day, you have this full coverage of the RBD, when you just take a random sampling of antibodies from vaccinated donors, suggesting that we were indeed making structurally, and functionally similar antibodies from the vaccine. And more importantly, we saw that classes 1 and 2 predominated, very similar to what you see in convalescent donors, which helped explain why we saw decreases in the neutralizing activity, especially against variants of concern that contain the E44K mutation. So we had this, within the first year, this understanding of the polyclonal response, monoclonal antibodies from convalescent donors and monoclonal antibodies from vaccinated donors, all showing, that the RBD was really the main target of our neutralizing antibody response, and that the vaccine was indeed inducing the same types of antibodies that could help, protect, people from infection. So in addition to looking at antibody responses, we also worked with a group out of Seattle to actually design the novel antibody ACE2 decoys. And so in this case, we took a structure of the ACE2 RBD, actually SARS-CoV-1 RBD structure bound to ACE2, and used that as a scaffold to understand how ACE2 recognizes its epitope. And they did the novel design to make these kind of, helical 3-helix bundle decoys, which had higher affinity towards the RBD compared to ACE2, and could outcompete it. And more so, through in the novel design, then I solved experimentally these structures. We saw that, computationally we could, get really close to actually what we see experimentally, and that the RMSD between our design and experimentally determined structure was less than 0.8 angstroms, with only a slight differences in enrollment conformation, showing you the power of computational de novo design. More so, these ACE2 decoys not only outcompeted, but they neutralized it with really high potency and was able to protect animals during the lethal challenge of SARS-CoV-2, right? Suggesting that these types of de novo design molecules could play a role in prevention moving forward, especially in high risk populations. And in addition, so after we designed new inhibitors, not only antibody inhibitors, but now ACE2 decoys, we also wanted to start to understand exactly what was happening as the pandemic continued to go along. And so in this study, we looked at the evolution of antibodies. And so now we had patients that were coming back 6 months post-recovery, or as I say, post-initial infection, and compare their antibodies that were proliferating, the B cells are proliferating upon the initial interaction with the virus, and how much they have matured. And so what we saw for the most part across the board is that antibodies and B cells underwent maturation, producing antibody or B cell receptors that had more somatic mutations and led to antibodies that had better, higher affinities towards the spike antigen. And in most cases, this higher affinity allowed these antibodies to neutralize, many different point variants, where their parental antibody that was isolated at 1 month were unable to do so. And so structurally, we looked at this and again began to realize that, at the molecular level, there were just small, minute changes, whereas the total surface area and barrier surface area of the antibodies were basically the same. Small changes within, at the residue level led to higher affinities, which then again allowed these antibodies to mature much better and retain their potency in the face of many variants of concern. And so we continue to see this now 1 year and 2 years down the road, where a lot of antibodies, that were first initially elicited and B cell clones that were initially elicited during the initial infection have continued to mature over time, leading to highly matured antibodies that they're more resilient to single point mutations and even some of the other variants are concerned. And this is observed not only in convalescent donors, but also among the vaccinated donors. So within a year, since March 2020, we were able to kind of characterize 5, different types of antibody responses, ACE2 decoys, as well as polyclonal responses, and begin to understand exactly how antibodies were maturing. So now, as we're moving into year 2 now of this pandemic, and I guess now starting year 3, Of course, we've all kind of switched now to say, okay, well, we have an effective vaccine against the current, spike. But as we have continued antigenic drift, how well are those vaccines going to protect us, as well as what's going to happen when the next, spillover event occurs, right? Are we going to have to go through this all again, knowing that, beta coronaviruses are found, zoonotic, coronavirus exists that have the potential to be, pandemic-like, right? So can we begin to identify cross-reactive antibodies that will protect not only in this pandemic, but also in future pandemics? So what I want to tell you now is three kind of, slightly longer stories of work that we're just published and work that we're doing currently in my group. And so, like I said, now we're shifting to the idea of, can we make a pan-coronavirus vaccine, or can we generate a pan-coronavirus therapy? And so I always talk about this because I think a lot of people, when they think pan-coronavirus, they don't realize how big the coronavirus family is, right? So for the most part, we're talking about the beta coronaviruses when you hear the term pan-coronavirus vaccines or therapies, and potentially alpha coronaviruses. But you also have delta and gamma coronaviruses, which are mostly found in livestock, but there are potential here for spillover events. But for the most part, we're looking at, the beta coronaviruses, and within the beta coronaviruses, the SARS-CoV virus lineage, right, which contains both SARS-1 as well as SARS-CoV-2, and a number of zoonotic ACE2 tropic, coronaviruses like LIV1 and SHCO14. So if you look at lineage B and kind of map, the conservation of all these different RBD sequences onto a structure and display it like this, which you can see that there are highly conserved patches that exist, corresponding mostly to that class 4 epitope in this cryptic pocket, as well as along the class 3 epitope, which is protected by glycan position 343. But if you look at where ACE2 binds, you can see it's a highly more variable, right? So we know that our initial and majority dominant response to SARS-CoV-2 infection is to these class 1 and 2 at the apex here, which is the more variable region. So we ask the question, can we identify, or can we generate antibodies or stimulate B cell receptors that target these highly more conserved sites? And so Alex Cohen, a graduate student in our postdoc in Pamela's group, and I worked to develop kind of this mosaic vaccine. And so I just quickly want to highlight this because this work, was published in last year, but we have a new preprint out describing its ability to protect now against challenge in not only in mice, but also in NHPs, in rhesus macaques. But the basis behind this work was that we decided we had this nanoparticle called MI3, in which we use a SPI-TAC spike capture system to display different RBDs on the surface of this particle. So this is a 60 more particle, so we could display up to 60 different RBDs. And so we made kind of this homotypic particle of just SARS-CoV-2, 2 different mosaics of each displaying 4 different, kind of like a quadrivalent mosaic particle, and then an octavalent particle displaying 8 different RBDs, making up all the different lineages of the SARS-CoV virus family. And so what we saw first off the bat was that, when we looked at neutralization, then we injected these prime and boost into mice, and then looked at the neutralizing response found in those mice. And so what we saw right off the bat is that, after prime, you had really strong neutralizing antibody responses across the board. But then after boost, you saw elevated responses. And so one thing about this graph I'll note is that the challenge virus, if it wasn't contained on the particle, then there's a red bar under that particle. So in this case, SARS-CoV-2 is on the homotypic, is on the 4A, and on the mosaic, but it's not on the 4B particles. Therefore, the response was muted after priming, but even after boosting with the 4B particle, which never saw SARS-CoV-2, you still see a neutralizing antibody response. So we did this with SARS-CoV-1. So none of these particles had SARS-CoV-1, but again, after prime, you see neutralization, especially with the mosaic. But then even after boost, even with the homotypic particle, we start boosting B cell responses that lead to neutralizing antibody activity. Then we also showed this again for SHCO14 and WEB1, which are zoonotic RBD or zoonotic coronaviruses that have the potential to spill over. All right, so across the board, we saw that our mosaic particles were able to boost or prime and then boost B cell receptor responses that can neutralize across a variety of coronavirus in the coronavirus family. But we didn't know exactly what was happening because, there is some sequence convergence or similarity between, for instance, Web1 and SARS-1. So we didn't understand whether or not this was just for the fact that this is so close to these two, or are we actually boosting and finding B cell receptors that are truly cross-reactive. So we went back to the human donors and asked that same question, saying, if we would go back and look for antibodies that can neutralize, SARS-CoV-1, especially in the US population in which we never saw SARS-CoV-1, could we begin to pull out antibodies that do that? And so we did. We pulled out two antibodies in particular, C118 and C022, which had broad reactivity against all the coronaviruses that we tested across all clades of the SARS-CoV virus family. In addition, these two antibodies also showed neutralizing activity, on par with some of our other antibodies that have been isolated to date. But again, you see it across all variants of concern at the time. This includes alpha, beta, New York variant, and I can't remember what 526 is the New York variant. I think this is the variant in California, but as well as zoonotic coronaviruses with one SHC-14, right? So that suggested that even after a SARS-CoV-2 infection, we are able to, make antibodies that have this broad cross-reactivity. So where exactly do they bind? So Claudia Jetty in the lab, a graduate student in the lab, who's actually defending tomorrow, 2 days from now, solve crystal structures in addition to the EM structures that we solved to highlight, exactly how these antibodies recognize their conserved epitope. So they target this highly conserved patch here in, like I said, that cryptic face on class 4. But they do so in a manner utilizing a long CRH3, which kind of reaches in and binds at an angle such that when you test whether or not ACE2 can bind, despite the fact that they don't actually have an epitope footprint that overlaps with the ACE2 binding site, they sterically hinder ACE2 and block ACE2 from binding using this different angle or this angle of approach. All right, so by basically using this long CRH3, which hits this highly conserved patch with a certain angle, you can now make these really potent neutralizing antibody responses, which is not to be said for all class 4 epitopes or class 4 antibodies, which is really showed here by graduate student Shannon Eastwin in the lab, right? And so I worked with her to characterize multiple class 4 antibodies that all bound to some degree this highly conserved epitope and showed cross-reactivity, but for the most part, they were non-neutralizing antibodies. And so when we look at it structurally, you can see that some of them bound at angles that did not allow for steric hindrance of ACE2, suggesting that steric hindrance of ACE2 is really important when targeting this ACE2 receptor binding motif. All right, so that's great, but that makes it really much more difficult to design immunogen that not only focuses in on a certain epitope, but also allows a certain angle of approach in order to achieve the high neutralizing activity that we want to see for protective antibodies. So with this knowledge, we decided to go look at a different epitope. And so in the study I did with a group at the Broad Institute outside of MIT, we looked at young donors from the Biogen conference, which is one of the first super spread events here in the US back in February of 2020. But we did a lot of work in this study, but at the end of the day, we identified 33 SARS-CoV-2 antibodies, 6 of which were really potent neutralizers. And that's kind of highlighted here, showing you that these 4 antibodies shown here, BG1019, 122, 425, and 715, all were able to neutralize to some extent, the wild type strain as well as the alpha and beta coronavirus variants, which were circulating at the time of the study, with BG1019 showing a very potent neutralization against authentic virus. We were seeing neutralization of less than 0.1 nanograms per ml. Right, so structurally, we characterized this antibody, and the structure was really, really unique in the sense that we took, again, looking at the variable regions of the Fab fragment bound to the spike glycoprotein shown here in gray. What we observed first off was that we didn't see any of these antibodies bound to an up RBD, right? So then when we look closer, we begin to realize that this, the epitope that was being recognized spanned across adjacent RBDs. in essence, stapling the spike glycoprotein close, never exposing the ACE2 receptor binding site. And so this wasn't the first time we observed this, right? And so in that Nature 2020 paper, we also observed an antibody that was able to bridge across adjacent RBDs. But in this case, it utilized a long steroid H3, which buried a hydrophobic tryptophan phenylalanine into this hydrophobic pocket at the base of the 343 glycan on the adjacent RBD. But when we looked at, this BGT19 structure, what we observed was the fact that, the main epitope being recognized was actually already around center on the 343 glycan, which again represents a conserved epitope, which I'll show you in a second, with the adjacent RBD being kind of captured or lidded by the light chain in the very non-specific interactions. So therefore, we even believe that we can engineer this antibody further to actually incorporate better, higher affinity interactions to make this even more potent. But what's really, like I said, caught our eyes, the fact that the epitope was really centered around this 343 glycan. And so, like I said, if you compare that to 144, whose primary epitope was in, overlaps in class 2, which is the ACE2 receptor binding motif, a more variable region shown here compared to a more conserved region shown here for the BG1019 antibody. All right, so having something that was not only potent in the sense that it staples the spike glycoprotein down, but also targets a highly conserved patch was really exciting. And in fact, what was even more promising was that the antibody that was going to the clinic, so this antibody from Beer Pharmaceuticals called S309, targeted a very similar patch. And in fact, this antibody S309 is still the only antibody that's being currently utilized. Well, I guess there's some new ones now, but for a time, it was the only antibody that was being utilized against the Omicron variant due to its ability to target this more conserved region and be unaffected by the RBD mutations in Omicron. Right, and so if we look, again at the S309 footprint and compare it to BG1019's footprint shown here in blue, we can see they target highly, very similar regions, whereas BG1019 has a little bit wider footprint. But more importantly, BG1019 still recognizes, the conserved enough epitope that can actually cross-react with SARS-CoV-1 with one and even SHCO14, albeit at a higher affinity. Measured affinity actually was around 360 nanomolar compared to 2 nanomolar for the SARS-CoV-2 RBD. But more importantly, when we tested in neutralization assays, we saw that BG1019 neutralized SARS-CoV-1 at a similar potency as S309, which is actually a SARS-CoV-2 induced antibody or SARS-CoV-1 induced antibody. And so we can see, like, not only do we have potent neutralization against SARS-CoV-1, but we also see it against SARS-CoV-2. And so why is it doing this? As I already kind of hinted at, is that, we know it can staple down this spike antigen. And so if we do a study, so we did a competition study to prove that indeed it wasn't allowing ACE2 to bind. And so again, if you look at how it would bind to a monomeric RBD, you can see that the footprint for BG1019 sits outside the RBD. And so what you see when you make a Fab RBD complex like this shown here, and then can ACE2 bind, you see clear binding of ACE2, which is this blue line here, compared to the RBD control. But when you ask the same question, you make now a spike Fab complex, we see no binding of, in the presence of BG19 relative to other antibodies like S309, which don't staple the spike glycoprotein close. So again, this led to us to understand this mechanism of stapling the trimer shut, preventing ACE2 from ever recognizing its epitope, is a really potent and powerful mechanism for neutralizing antibodies. And in fact, all the potent neutralizing antibodies that have been isolated to date show some type of similar mechanism in which they directly staple the trimer shut. Again, this is for these antibodies that have IC50s of less than 1 nanogram per mil. So in addition, we looked at escape from BG19. We weren't able to find any single point mutation. We did classical virology experiments in which we passaged BG19 in the presence of a replication competent virus in which the proofreader mechanism was removed. We identified some partial escape at position 441 and 339, but never saw full escape from BG1019. So this was great at the time, but of course, Omicron hit. And so we asked the question, can it bind Omicron? Unfortunately, no. And it's not due to the mutations at these positions, but actually to another pocket in which the CDRH3 tip fits into. There are some rearrangements around position 371, 373 in Omicron that make binding, that reduces the binding of this antibody. So we're working now to engineer BG1019 to overcome Omicron escape, and while also hoping to improve its breadth of coverage to have this kind of really unique neutralizing antibody that can staple the trimer shut and lead to highly potent neutralizing activity. But in addition to BG1019, there's this other story that was really interesting around this antibody BG122. And so BG122 represent a recurring class of antibodies corresponding to the 3-53 or 3-66 family of antibodies. So this is a public antibody that that was observed by us and many other groups. These antibodies in essence overlap with the ACE2 receptor binding motif and mimic ACE2 binding. And they're defined by not only these germline genes, but also by having a short CDRH3, which kind of sits in the on the surface of this of the receptor binding domain. So all these antibodies have a RBD of less, or CDRH3 of less than 11 amino acids. And so when you look at 3-53s that neutralize but had longer CDRH3s, what had been observed by other groups as well as us is that they have to bind with a different angle of approach to now accommodate this longer CDRH3, as shown here by antibody CoV-8-239, shown here in purple, and C144, shown here in blue, compared to the canonical 3-53 class 1 antibodies shown here in green. But in the case of BG122, that had a 21 amino acid long CDRH3, we saw similar binding as the canonical class 1 antibodies. And so we asked the question, how in the world is it binding when you have such a long CDRH3? And so when we saw the structure, we saw that, basically the CDRH3 was, just displaced in the sense that I couldn't actually model 7 of the amino acids of the CDRH3, which kind of goes against, for the, people that are, immunologists out there, where we know that CDRH3s are usually, bury around 50% of the antigen of the antibody epitope is buried against the CDRH3, right? But here's an example of an antibody that was raised in which it doesn't utilize the CDRH3, but really focuses on making sure that the germline coded interactions remain the same, and they just now displaces the CDRH3, and doesn't utilize it for anything. So that was really cool and goes against what we classically know about antibiotic responses. So I just kind of wanted to highlight the story because it's just something that we thought was really interesting among the antibodies that we had isolated. And so this was all work that led me through my postdoc. But as I left and talked with Michelle on my way out, I really became again focused on the N-terminal domain. Because I knew from my original study, we had N-terminal domain antibodies that were neutralizing antibodies. And, so I wanted to kind of focus back in on those. And so the research group at University of Washington, of course, published the first paper describing a lot of N-terminal domain antibodies in Iceland and showing there were kind of like 6 unique sites that were the target of NTD antibodies, showing the site 1 super site here at the apex and shown here in red, being antibodies that end up being neutralizing antibody responses. And so then when you looked at a lot of variants of concern, deletions or conformational or deletions that or mutations that led to conformational changes within the supersite basically made these antibodies targeting the site useless. And so a lot of the NTD antibodies were easily escaped by many of the variants of concern. And so we asked the question though, knowing that there's other, we saw from our polyclonal EM was there were dominant epitopes that were corresponding or correlating with neutralization within sera, within the sera of a convalescent donor. So I believe that if we were to sort with the NTD bait only and identify multiple antibodies, we could actually pull out new antibody classes that were indeed neutralizing. And so that's what we did. So we took, plasma from people 1 month and 12 month convalescence or 12 months after vaccination and kind of looked at their neutralizing activity and saw that for the most part, it was the same throughout. So you didn't see any increases in neutralizing activity, compared to what you would see with the RBD as these antibodies matured over time. They kind of fell into the, expanded the same V gene clones as we saw before. But when we tested, a series of these antibodies, we saw that 6 of them end up being quite potent, as well as very broad in their ability to neutralize across the different variants of concern. So what I'm showing you here are those 6 antibodies here on the y-axis against wild type alpha, beta, gamma, delta, Iola, and Omicron variants, as well as a synthetic variant called PMS20, which was highly similar or has some of the similar mutations as Omicron. None of them were cross-reactive in the sense they couldn't neutralize other beta coronaviruses, but the fact that we were able to identify antibodies that were able to neutralize across a panel of variants of concern suggested that these antibodies aren't targeting that major super site, which we know gets rearranged in a lot of these variants. And so we did a BLI mapping, and this kind of shows it here in that you had the same kind of 6 groups, site 1, kind of site 2, this large site 3 class. Then you had these 3 classes down here, 4, 5, and 6, which all corresponded to these neutralizing antibodies, right? And so here's the mapping of the competition BLI data. And then you have neutralization data shown here on this right y-axis, where again, you're looking at neutralization against wild type, gamma, this PMS20, as well as Omicron. So when you jump down here, antibodies that correspond to sites 4, 5, and 6, you can see now you start seeing, broad neutralizing activity. So I turned to structural biology using Cryo-EM and kind of solved structures from each one of these classes. The first being this antibody 1791, which belonged to class, I can't remember, class 5. And so you can see that kind of binds orthogonal to the site 1 supersite, very similar to this antibody S2L20, isolated by David Wiesner, actually utilized to solve the Omicron structure and Delta structure, but they didn't see neutralizing activity. So of the antibodies that were cross-reactive against the variants of concern, this was the weakest neutralizer, but we did see neutralizing activity, and it does have a slightly different footprint compared to SL20. But then we saw this antibody 1520, which corresponds to a class 4 antibody. This one bound the opposite side, orthogonal to the site 1 supersite, and it did so in a manner that positioned it in this highly hydrophobic cleft, which is the target of these kind of hydrophobic ligands that bury into this that actually play a role in potential antibody evasion. So kind of, density and molecular interactions are shown here, but what I really want you to focus on is this CDRH3. So it kind of, threads between the supersite beta hairpin shown here in red, and then this N4 loop shown here down at the base. And so you can see that, displaces these 2 loops, to really insert into this hydrophobic cleft. And so when we looked at other antibodies that have been isolated against this epitope, you can see that C1520's recognition of this epitope is somewhat unique. So these are other antibodies, but you can see we kind of, the CDRH3 kind of wedges in here, opens the gate, and doesn't really need to contact the beta site's hairpin, whereas this antibody S2X303, which also shows some cross-reactivity, but doesn't neutralize as well because it's up against that supersite beta hairpin. Same here, antibody 57 doesn't open the gate, engages the N4 loop, which again has many mutations found within variants of concern. And then this antibody P08 kind of, sits right in between in the open gate, but also buries a lot against the supersite beta hairpin. So from this structure, we understood that, engagement of this epitope to be potent and cross-reactive requires that you open the gate, but don't actually engage that supersite beta hairpin where a lot of the mutations fall. And then finally, there was this antibody C1717 from class 6. And this one was really, really, really unique in that it bound, kind of proximal to the viral membrane, which would be here at the base of the spike antigen or spike glycoprotein, and did so in a manner in a position that places it really close to the fusion peptide machinery. And so we believe, that, part of the mechanism of these antibodies is actually to sterically hinder processing or conformational changes that leads directly to fusion. And we're currently testing that hypothesis in my lab right now. But overall, by binding this, epitope at the proximal site of the spike antigen, you miss a lot of the, mutations that are found in variants of concern, including those found within Omicron. So at the end of the day, this is what 2 years now of COVID-19 structural biology looks like. We were able to really define RBD specific antibodies across convalescent donors as well as vaccinated donors, and understand those antibodies as they evolve over time. We were able to look at and design ACE2 decoys, as well as begin to understand cross-reactive antibody responses that could be utilized in protecting us and making new vaccines, antibodies that we could improve as therapies to help protect us in the emergence of other and new variants of concerns, as well as antibodies targeting epitopes outside the NTD, which again will help us as we continue to see antigenic drift that's occurring. One study I didn't talk about, but was just published, was actually targeting many different novel epitopes that were identified using mRNA phage display library, which again allowed us to rapidly engineer and make new antibodies in the face of many different variants. And so in this study, we started with in the summer of 2020, and by the time it got published, we had made 4 new antibodies really quickly, corresponding to alpha, beta, the California variant, and then the New York variant as well. So with that, I'll end and, just want to say thank you, of course, to Pamela Bjorkman, who was my postdoctoral advisor. She mentored me throughout, my time in her lab, allowed me to get where I am at now. Many people within her lab to thank, a lot of graduate students and postdocs, staff scientists, and research assistants that I had the pleasure to work with. But again, highlighting Anthony West, who was a bioinformatics person, did a lot of work on understanding V genes. Claudia Jetty, who was a graduate student, Morgan Abernathy, who was also a graduate student that helped solve a lot of these structures with me. And of course, Beth Huey-Tubman, who worked as my technician, especially at the beginning of the pandemic when we were receiving samples, and Caltech doesn't have a hospital, we don't have anything. So, working through protocols to understand how to handle human plasmas, was not an easy task, nonetheless, but we were able to do it with a lot of help from Beth. From Rockefeller University, Michelle Nussensweig's lab, especially Davide and Vinci, who helped characterize me in these antibodies to talk about today. Paul Binash's group, Franca and Theodore, as well as Iska, who helped do a lot of neutralization assays. The BG219 stories with our collaboration at the Broad Institute with Romnick Xavier's lab, and Johannes was the main driver of that work. Our people at the BioHub, John Pack, our facility directors that kept our microscopes up and going, Andre and Sonia, being-line scientists at Stanford's SSRL, and there are companies and other collaborations that we had throughout the pandemic. To all of our funding, and most important to the volunteers and all the scientists who've worked really hard to make sure we get out of this pandemic and get back to, quote unquote, normal life, which, hopefully we'll get there. And then I just want to highlight, yes, that now I've started my lab, now I guess 9 months into it at Stanford University. I'm part of the new ChEM-H Institute as part of the Department of Biology, brand new building, this brings together a diverse group of disciplines, from live, physical, and clinical sciences, as well as a diverse group of people to drive, innovation in human health. And so I'm really lucky to be able to now train my own graduate students, highlighting Gina and Adonis here, who actually just won the NSF Graduate Research Fellowship today. So I'm really excited for them. But our lab is growing, we're recruiting postdocs, and thanks to our funding to get some of the work off the ground. So with that, I'll stop and happy to take on a couple of questions.
Thank you so much, Chris. It was a really great and interesting talk. It's a time of Q&A session. If you have any questions, please raise your hand or type your questions. Yes, Patrick.
Hi, Chris, really interesting talk. And I'm going to go tangential to this. the alternative, potentially for acute antibody therapies is not to target the viruses, but the ACE2. And I think you can actually get antibodies that will bind to the ACE2 without inhibiting the enzyme function. So what do you think of that approach and where it sits relative to therapeutic antibodies for acute treatment?
No, I think that's a great approach, and you're right. There was a paper not too long ago, describing an antibody actually targeting ACE2, and it showed that it was protective. But to your point, right, if you target ACE2 in a way that actually doesn't inhibit ACE2 function, then you should be able to achieve, neutralization of the virus, despite whatever mutations that the virus carries within these different variants of concern. So yes, I think that is a great approach. I know people are working on that approach, and so I would say it's definitely something useful that you can do. Obviously, the concern would be, ensuring that you aren't inhibiting ACE2 function, obviously because of the fact that ACE2 is a really important enzyme. So I think if you can, really show that you're not going to have kind of these autoantibodies or other antibodies that would then, affect ACE2 within a clinical setting, then it would be a great way forward to make an antibody therapy that is basically inescapable.
Yeah, obviously it's not a vaccine approach, but as an acute therapy, yeah, very complementary to the sort of other work that you're doing.
Yes.
Thank you.
Mm-hmm. And to that point too, like I'll even raise you one further, which is the use of kind of these from de novo design, these ACE2 mini binders or other things that we know we can formulate into a nasal spray. And so actually now you're delivering these small molecules in a setting where you're going to actually get them into the lungs and protect, the ACE2 that's being displayed on or bind to ACE2 on the cells of these lungs. So I think that's even a more powerful approach, especially, for people that are really at risk and may need it. Yeah.
Sorry, again, if you're not inhibiting the enzymatic activity, because obviously it's protective in lung disease and so forth. Yeah.
Yeah.
And any other questions from guests? Lester.
Hi there. Great talk. I suppose I just had a quick question. I suppose when I think about this spike protein, some of the papers that I've seen peripherally sort of show it maybe in multiple conformations, and part of its function is to sort of go into this up position. But all the structures that you've really shown are really the spike protein with maybe the RBD up or down. Do you think there's any antibodies that are able to bind it when it's in a vastly different conformation, or is it sort of too late then that you've already engaged with the ACE2 receptor and it's sort of game over?
Yeah, no, that's a great question. And so we've thought about this a lot. And so, of course, one of the hallmarks of the mRNA vaccines, even Johnson & Johnson's, Advax, vaccine, it's a 2P mutation to kind of stabilize that pre-fusion, state. So what you're talking about and describing is this post-fusion state where it kind of elongates, and then will, of course, be leading to this, insertion of the fusion peptide into the host membrane. So, when you look at those antibodies, and so we haven't, but I know others that have looked at those antibodies. They tend to be antibodies that are non-neutralizing, because to your point, like it's already, engaged ACE2 and led to this, this state. And so these non-neutralizing antibodies actually can dilute neutralizing antibody responses. But it's not to say that these antibodies might not have other functions. So they could potentially mark virus or, spike protein that's now expressed on the surface of the cell for destruction by other, immune cells through their FC functions. But, what's really interesting is if you look at I think it's Sinovac vaccine, it's a live attenuated vaccine in which they use this reagent that is known to actually promote or shift the equilibrium to that post-fusion conformation, this elongated conformation. And that vaccine is actually less effective. And so we, hypothesize the reason it being is because you're making a ton of antibody responses now to spike that's being displayed as this post-fusion state versus the pre-fusion state that you would want to target to prevent entry. So I mean, there's possible functions for those types of antibodies, but from a vaccine or preventive standpoint, I do think you want to target the pre-fusion state to prevent injury.
Yeah, cool, thanks.
Mm-hmm.
Thank you. Any other questions? I think it's all from the guests.
Sorry.
Yes.
Since nobody else is asking a question, I found that the sort of new class that the last set of antibodies you're talking to, that bind in the different epitope near the fusion domain, quite fascinating. What's the sort of relative affinity of those antibodies at this point in time, and are they where you might want them to be, or are they your potential targets for affinity maturation and so forth?
Yeah, so that's a great question. So in this case, these antibodies are targeting, or this antibody in particular is targeting the N-terminal domain, but it kind of also overlaps with, as you see here, the SD2 domain within the S1 subunit. And so these actually have a pretty high affinity. I believe the affinity for this antibody was around 10 to 20 nanomolar. But so yes, it can be improved, but it's already pretty high affinity. But now there are some other antibodies that are being isolated that have really this pan-coronavirus, activity, because they are targeting the fusion peptide, right? So when you start targeting domains within the S2 subunit, that's kind of what people are focusing on now, because we know antibodies targeting the S2 HR2 stem helix down here, as well as potentially those around the fusion peptide machinery. These sequences are highly conserved across all alpha and beta coronaviruses. And so what we're seeing, though, is that while you have a high affinity, most of these epitopes are very cryptic, and it may require other conformational changes to expose these epitopes, on the surface of the virus itself, not just the peptide, right? So a lot of these antibodies are being fished out by peptides, and they bind very well, but the presentation of these within the pre-fusion spike trimer is Incompatible, right? And so I think what we're seeing now, at least from our own data and data from from David Wiesler in essence, we see that when you have antibodies that mimic either ACE2 binding or have ACE2 binding leading to some conformational changes, you can now start exposing some of these new epitopes, which have the potential to be the target of these pan-coronavirus types of antibodies. So I think if we can find these S2 epitopes or find regions that are highly conserved, and start with a library, and then make these antibodies and hopefully enlist them in a vaccine, that would be great. And so there's room there to try to really improve the affinities. But I think all these antibodies, for the most part, even the BG219 story I was describing earlier, these are all antibodies isolated with very few somatic mutations. So we know we can improve their affinity, and the affinity improvements actually make them more resistant to single point mutations, which could be the difference in keeping them from being resistant to the virus.
Cool. I think somebody else has a question, so I won't do my follow-up, and I guess we're getting close to time. So,
yeah.
Don G, back to you to.
Yeah, it's 11 now. So I close it first.
Maybe let Peter, because he's had his hand up.
Oh, okay. Yeah. Here. Sorry.
Oh, that's all right. Thanks, Chris. Just curious. Just want to get your opinion on, I guess, kind of more recent vaccine technologies, just focusing on the RBD portion of the spike protein, considering that a lot of your work showing that you're getting kind of effective cross-reactivity of antibodies targeted away, like outside of the RBD domain. So just curious what your opinion is on like kind of focused targeting of just the RBD domain of the spike.
Yeah, so no, I mean, this is, what work we had started during my postdoc, and this is currently being led, like I said, by Alex. So in this case, we only utilize RBD in the sense, again, focusing in on just preventing the SARS-CoV virus lineage. And so, I mean, if you just focus on making a kind of a pan SARS-CoV virus vaccine, then utilizing the RBD works because you have such high sequence conservation within the RBD. And we kind of show that here, right? Where you can make particles of just the RBD and get neutralizing responses in vitro. And like I said, there's a new preprint just went online where Alex is the lead author from Pamela's group, where we actually took these same particles, immunized twice, and then challenged animals, not only mice but also rhesus macaques. And you see again very protective. You saw protection from a challenge in a challenge study. So I think if you're narrowly focused on just a single lineage, then RBD specific epitopes are fine. But if you really want to make a larger pan coronavirus vaccine or antibody therapy, then I think that requires now targeting epitopes that sit outside of the RBD itself, such as those targeting, not even NTD, because again, NTD is very variable, but again, targeting those in the S2 domain of the spike protein.
Yeah, thanks. So effectively, good for now, but like, get the whole spike, and get the whole spike, and you potentially are protecting for kind of future variants and things down the track.
Yeah, I mean, I think that's been the hardest part, because we know that we have all these other endemic coronaviruses, right? And so we can utilize, like, for instance, beta coronavirus OC43, endemic virus. We can isolate antibodies against, again, the ChEM-2 helix, but are those really protective antibodies? So now I think the focus is can we find these highly potent neutralizing epitopes within S2, that can be the main target of a new antibody focused response, now generate, that in the vaccine. So that way, yes, we can not only prevent against SARS-CoV virus family, but now also, MERS and all these other beta and alpha coronaviruses.
Cool, thank you for that.
Mm-hmm.
It's 11:04, so I will close this session. Thank you, Chris, and thank you for attending our ChEM-MP seminar, and see you next month, everyone.
Thank you, guys.
Thanks, Chris.